if
This
possible.
conference will now be recorded.
Yeah. Okay. So if you so the title of my talk is BacteriaBot, a novel drug delivery system for oral drug delivery. Okay, or for oral administration, whatever you want to say. So before I go into that, let me just tell you what all the delivery agents, all the delivery routes we have, and then I'll come back then on different problems and all. So the delivery routes through which we take drugs are many, like oral, it can be subcutaneous, intravenous, intraperitoneal, intramuscular, and so on and so forth. And every route has some advantages as well as some disadvantages. And the choice of drug or the choice of the route, it depends on the drug based on its property, whether we want a drug to be metabolized uh, or we want the drug to be intact, go for the action, whether the drug is stable in presence of gastric acid or it's not, or whether you want a very slow release or you want a fast release in the blood, and very other parameters are there based on what happens, your choice of drug administration route varies. Okay, so we'll be talking mostly about oral route, how it changes. But before we go into that, uh, nowadays what we are running out of drugs, and it has many reasons. Reason includes manufacturing. Some drugs are really good but difficult to manufacture. Supply and demands are there. We have a lot of half of those are unknown reasons actually. And but one of the problem is with the 10% of our drug is it got discontinued because they have low GI tract absorption rate. And when you apply those drugs to some other route, through some other route, it causes problem, a lot of problem. Like it causes anaphylactic shock, or it needs a metabolic effect before it can go, so it doesn't go through that metabolism effect and all. And sometimes it might be you need a drug to be released slowly in the blood, but if you inject it or take it through any other way, it goes in the system very fast. So for those reasons, 10% of the drugs actually what we have in the market, we cannot use it, although they are very potent. Now, if you compare those drugs, what you have, you see majority of them are antibiotics, and some of those are chemotherapeutic agent, and many other diseases are there like cardiovascular and all. So, and especially antibiotic, if you know the WHO has predicted that by 2030 we'll be out of all the antibiotics what we have right now, because most of the most of the bacteria, microorganisms will develop resistant to the existing antibiotic by 2030. So we are really in problem or in a verge of ending of all our antibiotics. So, and not only for antibiotics, there are other diseases which where we need this kind of support. We need new drugs, or else because making a new drug takes around like 15 to 20 years, at least 10 years to come to the market, unless you have a COVID situation, obviously. Because of the COVID situation, some drugs we are expecting to come to the market within 1 to 2 years, but that's not the general scenario. You don't get an approval of a drug within 1 to 2 years in general. So if you want to avoid that, because if you think about 2030, we don't have even 10 years. So if you need to avoid that situation, what we need to do is we need to get some way out to use or make use of our existing drugs which we are not using anymore. And this method may help us to do that. So now let's talk about a little bit on oral route and what actually the problem is. So oral route is most common route of drug administration. As you know, almost like 85% to 90% of the drugs what we take is through oral route. It's very convenient. You can take it, you can do like self-administration, self-administer the drug. You don't need any injection or something which is not painful. You can take it like time to time. Oral drug absorption rate is low, so it don't go at a very fast rate or don't increase the blood concentration of drug at a very fast rate. So what happens is basically you see a kind of sustained effect of drug in the blood. For many reasons, for many drugs, that's very important, that you don't get the concentration at a very fast, or you don't get a see concentration max in the blood at a very fast rate. So for many drugs, that's very important. And then it's very economic, and especially drugs which are highly soluble and polar. It's not exactly polar, somehow polar, and easily can permeabilize the GI tract wall, can be easily taken by this route. But the majority of problems, so what we feel is the drugs which are like little less soluble, or they are having low permeability, in those or both. If you are taking those drugs through oral route, what will happen is mostly say 5 to 10% of the drugs will get absorbed through the blood or through the intestine, because intestinal absorption happens inside your stomach and small intestine. Once it reaches to the large intestine, you don't get any absorption. Almost like no absorption happens through large intestine. So the window what that drugs get is like around half an hour. It varies from drug to drug based on your solubility, based on your binding interactions and other things. It varies somewhere between like 15 minutes to maximum 1 hour. That's the window it's get to get absorbed from the oral route or GI tract to the blood. Now, that window is not enough for many drugs which has low solubility or low permeability through GI tract, and only you see around 3 to maybe 10% absorption of that drug from the blood or into the blood from the GI tract, and that 90% of the drugs actually comes out through the feces. Now, why is that important, or why is that problematic? I would rather say the reason is, if you're 90% of the drugs is coming out through the feces, what happens is the drugs directly goes to the environment and mixes to the environmental microbiome, right? What happens as a result is you develop the microbiomes which is already present in the environment. They get exposed to the drug and they get resistance. Especially when I'm talking about antibiotics. Not only that, that drug can go to the water circulation, that drugs can go to the soil circulation and all. So the environment, you can see a biomagnification effect on the environment because of these drugs which is coming out through your feces, which is not been absorbed in GI tract. That's one of the biggest problems. Other problems are there is specifically people who has irritable bowel disease, or say some kind of infection or ulcer or tumor in GI tract. When you're using that much excess amount of drugs, say 90% of the drugs you don't need, so it's going through your going out and actually coming to your large intestine and staying there, and that can cause a lot of irritation, a lot of inflammation to those patients who are already suffering. So for many patients who has this kind of problem, they cannot take drugs through oral route, they need to choose some other drugs. Further, it's even not economical, right? You are losing 90% of the drugs, so it's not economical. So what's the way out? To solve this problem, what many researchers has done, they have made excellent nano formulations and chemical modification, obviously. Now, I'll rather talk about excellent nano formulations or sustained release formulations. because that's very important. That's give you a release of the drugs happens very slowly and very efficiently, which allows the drugs to be absorbed from the intestine. Okay, when you make a sustained release formulation. I know Dr. Deobrata Joshi has made a wonderful paclitaxel formulation. Dr. Deobrata from your university has made a wonderful solid lipid paclitaxel formulation which shows a sustained release and very effective in lung cancer. Lung cancer, right? So these kind of formulations people have made, but when it comes for the actual application, there is a problem. Problem is your formulation might be very good, but whenever you are taking it through oral route, it is going to stay there inside your stomach as well as in your intestine for again that maximum 1 hour window. And if that window will not get increased, because of changing of solubility, you might get absorption better, but you cannot absorb completely. If you can increase the window of that absorption, you can achieve a lot of the drugs to be absorbed by the blood through oral route, which otherwise cannot possible. So that's what we're working on. So what I made is basically, you must know that our body is like, even our body, half of our cell which is present inside our body are bacteria, and our intestine and stomach or the whole GI tract is full of bacteria. They help us for digestion, they help us for maintaining bowel secretion, they help us for maintaining the pH of the stomach or intestine and everything else. So, and if you get rid of those bacteria, we might not be able to live how we live, right? And you might know as well, so when people get stomach infection or something, many times doctors gives you bacteria. You have to get good bacteria in your stomach and your stomach will be fine. So this bacteria has a very important role to play, or this microbiome has a very important role to play in maintaining our metabolism, right? So, and this bacteria goes and stays inside your stomach or inside your GI tract. Not stomach. Stomach has a lot of acidity, so acid. So there are some bacteria in stomach, obviously, but mostly it goes and stays inside your intestine, small intestine and large intestine. And obviously, small intestine helps us for absorption. So we'll be more discussing about small intestine than large. This bacteria goes, sticks, and stays there for many days. So what I did is we took some of the intestinal bacteria and we used that bacteria as a vehicle to deliver the drug. Now, bacteria, if you put the drug inside the bacteria, it's first of all, it's a very difficult job to put the drug inside the bacteria. You need very difficult formulation, which increase the cost, increase the technological difficulties and all. And secondly, even if you put the drug inside the bacteria, you don't know whether the drug is going to get released, because the release of the drug is very important in the intestine. So what I did is, instead of putting the drug inside the bacteria, we made the bacterial surface as a drug vehicle. So what we did is we poured nanoparticles on the surface of the bacteria, which has very tiny pores, mesopores, and which can absorb drug and release drug, show sustained release effect. And these drugs, we did it in living conditions so that these bacteria can go and bind over there, and over there means inside your stomach or inside your intestines, and can release the drug for a very long time. So our objective was to increase the time span, the window of which the drug gets absorbed. So that's what we try to do over here. So that's what I'm going to explain in next half an hour or so. Please feel free to stop if you have any questions. I have no issues. Okay. So what I was talking about is we took this bacteria Lactobacillus reuteri. We also did it with Bifidobacter KSE. So you can take any probiotic bacteria for these kind of purposes. So normally these bacteria look like something like this, and if you put this nanoparticle on the surface of the bacteria, you can see this surface is smooth, but after putting these particles on the surface of the bacteria, the surface has become very rough in nature. This is a FESM image, which is Fourier transform scanning electron microscopic image, right? So, and then a very small portion of that is on the surface of the bacteria. You can see the particles. These particles, what I was talking about, are tinier particles. It's around 15 to 25 nanometer in diameter, which has a pore size between 2 to 3 nanometer. And these are like sponge balls, little sponge balls, which can absorb a lot of drugs at a very fast rate when you put a drug concentration at very high rate, and it can release the drug slowly when drug concentration is less outside. And again, you can change the polarity. You can do a little tweaking in the polymer. So we have used certain polymers over here to make these particles. The polymers is the data I'm showing is with the chitosan particles, but we have made other polymers and we need some modifications as well. So if you do a little tweaking on the polymer charge, you can actually accommodate a huge lot of drugs inside this vehicle. So these are like microcapsules. Now these bacteria are microcapsules, and if this bacteria can go and stick to the intestine, what will happen is these bacteria will release the drug from their surface inside the intestine. And since they're bound to the surface of the intestine, the release kinetics would be very high, very, very long, and intestine will get, or stomach will get enough time to absorb all the drugs which it needs. So for that, okay, so I'll come and explain the data, and then I'll show how it's actually affects. So the first point was here. So once we did this this encapsulation of the nanoparticles on the bacteria, we are calling it bacterial boat, by the way. That's the term we're calling this nanoparticle encapsulated bacteria. We have given the term as bacterial boat. Bacteria as a vehicle. Boat means which carries a lot of cargo, right? So bacteria, cargo transferring bacteria, which is a bacterial boat. Now, the first question you want to ask is, okay, great, you have put some nanoparticle on the surface of the bacteria. So does this bacteria actually alive after this? Because to stick to the intestine or to the stomach, the bacteria has to be living. If you take a dead bacteria, it's not going to be absorbed. I'll explain why it's not going to absorb, but take that point first. So what I did, we actually did simple bacterial growth analysis. You know bacterial growth analysis you're familiar with. You can culture the bacteria. We for this particular bacteria, we used MRS media. You can culture the bacteria and take time to time OD, and you plot the OD based on the, and you when the OD will increase, you know the bacteria is growing. If the OD is constant, you know the bacteria is not growing. So at the initial phase, what happens is, this is control bacteria, control Lactobacillus reuteri. When we put these particles on this bacteria, what happens is, what we found is a sharp decrease of growth rate. You can see these brown line. Brown line, it shows EMT means empty. Bacterial boat is the nanoparticle encapsulated bacteria, right? So empty bacterial boat showed little less at the initial phase of growth. And then we tried with a very toxic drug, which is 5-fluorouracil. which actually used basically capecitabine, which is its orally applicable derivative. But we use this drug to load on the particles, and we saw a further decrease of growth because this drug is quite toxic. We also say we didn't load any particles. This is the green curve shows only lactobacillus with the 5-fluorouracil, and this curve shows lactobacillus with 5-fluorouracil, Bacteriobot loaded with 5-fluorouracil. So what C is only lactobacillus 5-fluorouracil also shows decrease of growth. And when you do it on the Bacteriobot, since Bacteriobot formation itself causes decrease of growth, you find a quite decrease of growth at the initial phase. But when you actually keep this experiment going, this is 168 hours data, you can see the bacteria has reached till the maximum growth level for all the curves. For obviously drug-loaded bacteria took longer time. Now this is actually very advantageous to us, because this picture can also show you that if you see the diameter of the bacteria, this is just 12 hours old bacteria after loading. So if you see the length of the bacteria, here the total length of the bacteria is around 1 micrometer, right? The control bacteria when nanoparticles are not loaded. But when you load these nanoparticles on the bacteria, you see, if you see the diameter, the length and width as well, what you see is the length has increased almost like 2 to 3 micrometer, around 2 to 3 micrometer, which just shows that bacteria is not dividing, is just elongating. They are metabolically active, they are live, but they are not been able to divide. The reason is we have done a modification, covalent modification on the cell wall. I'm not actually sharing right now the protocol, exact protocol how we did it, because this is although we have already applied for the patent, but this is right now we're actually writing the manuscript. So and we are also trying to do clinical trials. So before we do that, it's very difficult for me to explain everything. But take my words that we did some chemical modification, covalent modification on the cell wall, so that the bacterial nanoparticles on the surface of the bacteria remain very stable and they don't fall off very easily. Now, when we did that, it actually gave us an advantage. It reduced the growth of the bacteria. Why it's important? Because it will make the particle stable on the bacterial surface. If the particle is not stable, or if the cell wall is very easily fragile, it keeps on dividing, what will happen? You will see a very fast dilution of the particles, and the particles may fall off from the surface. But since their growth has decreased, so the particles are actually stable. Now, we also further confirmed the metabolic activity of the bacteria using some other parameters. So we are dealing with lactobacillus, which is known to produce lactose. Bacteria, and you can see the empty Bacteriobot or Lactobacillus reuteri, it should be L. reuteri instead of S. reuteri, or 5-fluorouracil loaded Bacteriobot all have more or less same lactic acid production efficiency. With Lactobacillus reuteri probably has a little more, but they are not that significant. So that means they are metabolically active and their metabolism rate is very similar. Okay, so is there any question so far? Am I clear what I'm talking about right now? Anyone? Shall I stop for a minute so that you may ask me some question or something? Anyway, if you don't have a question, let's just continue. Yes, there's some question. Anyone?
No, Dr. Chaudhary, really I'm extremely sorry. I had some internet issues and that is why my whole thing fell off. I am Dr. Sengupta.
Okay, hi, Dr.
Institute of Pharmaceutical and Technology. I had these issues, but anyway, generally we tell our students that whenever they have questions, they may write in the chat box, and maybe at the end or whenever they write.
Dr. Sengupta, your voice is cracking. I'm not been able to hear.
I know. This net problem, internet problem I'm getting. Okay, please continue, sir.
Dr. Sengupta, I'm really sorry. I'm not been able to hear a single word from your side. Anyway, I think your point was that your students generally ask questions after the talk, right? So I'll just continue then. Okay. Sorry, I'm not been able to listen to you, but I hope you can listen to my voice. So the next question comes. I'll come on one more thing, but next question comes whether these particles are stable in different stomach pH and stomach and or GI tract pH and GI tract environment. Now, the pH of the GI tract varies from pH 2 till pH 7.4. Like when the intestine is very close to the GI tract or intestine is very close to the stomach, the pH of the system remains around intestinal pH, which is pH 2, and then it keep on growing, and the average pH of in the intestine is around 6.4, and when it reaches all the way to large intestine, its pH remains around 7.4. So we work in all these three pHs to check the stability. And we used simulated intestinal fluids as well as simulated gastric fluids. So that means we had all the salts which is present inside the simulated inside the intestinal fluid, as well as inside the gastric fluid, as well as the enzymes like you have pancreases enzymes in intestine, and then you have amylases and other things in your stomach. So we have tested in presence of those enzymes as well as the salt, and we check the stability of those particles till 72 hours. So this is for pH 2. I'm just showing the data. So at the beginning of around pH at 12 hour, it's okay. There are some distortion is always there. So I gave an overall scenario. So then 36 hours, 48 hours, and till 72 hours, most of the particles looks very stable. When I went from pH 2 to pH 6.4, we still found the same pattern. The particles are very stable on the bacterial surface. What you also see is probably the swelling of the bacteria, swelling of the particles. If you compare particles which is present, if you compare particles which is present over here and versus here, this particle might look like a little swelled because you are putting the particles for 72 hours inside the fluid for a long time. Because of the fluid, they are swelling up. And this is for pH 7.4. And what interesting thing you can also see is basically expression of these. What are these? These are basically biofilms. Now, why this biofilm is very important? You can see it. This is after we started the experiment. After putting the particles, we waited for like 48 hours before we started the experiment for pH 2. The image you shows it's before starting the experiment at pH 2. You see a lot of biofilm production is also there, and then these are with the fresh cells, so you don't have any biofilm over here. If you wait, it secretes a lot of biofilm, and this biofilm is very important. Very important in the sense, the reason bacteria goes and stick to your intestine is because of this biofilm. These biofilms are made up of carbohydrates. and intestines are made to absorb the carbohydrate. These carbohydrates are it's been absorbed by our intestine as well as is being degraded by our intestine. So once the bacteria start secreting this carbohydrate, what intestine tries to do is intestine tries to take that up. So, and these carbohydrates are basically linked to the cell surface. So it goes and they keep on secreting the carbohydrate, and while doing that, it goes and stick to the intestinal surface, because intestinal surface is constantly taking up the carbohydrate from the surface. So these interactions, this mutual understanding of secretion of carbohydrate or secretion of biofilm and absorption of the biofilm allows this bacteria to stick over there. So for our experiment, this biofilm production was really important. If it can't make biofilm, then it's of no use. So we checked the production of biofilm efficiency at different pH, and what we found is they can produce a lot of biofilms even at 24 hours. See, this is normal lactobacillus reuteri and this is Bacterobotes. What you see is if this is only an MRS media, and this is in presence MRS media along with definitely simulated intestinal fluid, and this has pepsin because pepsin is important, which is present in gastric juice, in stomach, which is a protein degrading enzyme. Right. So we also we saw that in both conditions they produce a lot of biofilms, and with increase of enzyme concentration, obviously the biofilm concentration decreased, but that's good enough to so that it can go and absorb on the surface. If it would be absolutely zero, then the absorption rate would be much less. But if it can, its absorption rate is if its production rate is high, then that can go and stick. So still 72 hours, they are producing enough biofilm which can allow them to go and stick to the intestinal surface and stomach surface. And the production of biofilms actually increased with increase of pH. So in stomach, the system is stable, but if you compare stomach versus your intestine, this is even better, more stable in intestine. So I'll come and show you how it goes and binds to the intestine and stomach. Sorry, my slides are not moving. I'm sorry, my slides are not moving. Can you please help?
Yes, sir.
Yeah, thank you. Okay. So again, so you can see my slides, right? I'm on the next slide. So again, this biofilm production actually depends on the how much chitosan means the we are talking about the polymer we have used to make the particles on the surface. If you put less amount of chitosan, you see what you see is production of these particles at less. This is at 24 and 48 hours data. But when you increase the chitosan concentration, this is at pH 7.4, you see a huge increase of biofilm production in compare with control. Okay. Now this is your chitosan blank. This is only lactobacillus reuteri at 24 and 48 hours, and then when you increase the chitosan concentration or time, you see a high biofilm production. As well as at the same time, if you see the microscopic image, the size of the bacteria clearly shows, and the secretion clearly shows that there's a lot of biofilm product is being produced, but cell is not dividing because there is no evagination in between. It's just becoming elongated. Okay. Now, so as I mentioned, is it actually true that it goes and binds to the intestine and it can show release drug from there? So to test that, what we did is we loaded rhodamine in the empty bacterial boat and allowed that rhodamine loaded empty bacterial boat to go and interact with freshly separated goat GI tract. And what you can see is basically absorption of those rhodamine loaded bacteria on the stomach lining as well as on the intestinal lining. This ring shows basically the alveoli, where around this alveoli you can see accumulation of this bacteria. So the bacteria actually is going and binding to the alveoli, and then these alveoli are actually responsible for the absorption. So that means it's a very good situation where you can, if you can load the drug, it will go and bind to very close to the alveoli, and it can directly be absorbed. Drugs can directly be absorbed from that particular place. So that's what we got from our microscopic studies. Then we further try to confirm. So this this curve also shows basically the rhodamine loading efficiency. With empty bacterial boat, you have the maximum rhodamine loading efficiency. Okay. Now we also did same analysis, that whether is this bacteria actually binding to the intestinal and gastrointestinal tract or not. So this particular slide, the picture number B shows FSM of intestinal tissue, of goat, and then we added bacterial boat on the surface. What you see is those attachment of those bacterial boat. You can see those particles on the surface of the bacteria as well in some cases. So this is just some more elaborative images. So this is basically the stomach and this is intestine, where you can see those finger-like projections. These are for using goat intestine and goat stomach. And so when we applied those bacteria, you see the absorption. This is on the intestine and this is on the stomach. So you see the absorption of the bacteria on those organs. Okay. Now, so what? Now bacteria is gonna is showing better binding. Whatever you make, it's alive. It can go to intestine or stomach. It can bind over there and stay over there. So what? To test whether this is actually helpful to cure some disease, what we used is basically a drug, a quite popular drug, 5-fluorouracil, which is very so if you know what is a 5-fluorouracil, it's a basically uracil, where at the 5th position you have a fluorine moiety. Now this fluorine moiety allows the, now uracil, you know, it goes and incorporated inside the RNA, right? But when you 5-fluorouracil is more similar to structure of thymine, because here in thymine, what you have, instead of this fluorine, you have a methyl group. So it can go and incorporate it inside the DNA. And when it gets incorporated in the DNA, on the very next cycle, the replication enzyme cannot recognize it anymore and it dies. The cell dies off. Also, it inhibits some enzymes known as thymidine kinase and all. So overall, the purpose of this drug is to kill the cancer cell by inhibiting replication. And this is also known as suicidal inhibitor, because when you apply the drug, the cells absorb it, it incorporates, and then it dies. This particular drug is very effective against various types of cancer: colorectal cancer, breast cancer, sarcomas, liver cancer, and prostate cancer, etc. But the main problem of the drug is you cannot use it through oral route, because it has half-life period of only 8 to 20 minutes, and that also varies upon the patient. The patient has a high metabolism rate or something, it has half-life of around 8 to 10 minutes. That's it. And that time is not enough for the drug to be absorbed and show effect, because actually that time allows only a very small amount of drug to be absorbed. Although its solubility is fabulous. So what we do is, for oral delivery, we take a prodrug, which is capecitabine, where you have a ribose and then an amide chain attached to it. This increase the drug, the half-life of the drug from 8 minutes to around 30 to 45 minutes, which is good enough for the drug to be absorbed. And this drug is quite popular for use for different type of cancer. So we also try to see whether whether these drugs actually going and getting incorporated in the particles and showing stability. So we did isothermal calorimetry studies, and we found very high negative del H and del G value, which shows the particles goes inside and the interaction is quite stable. So that means it will show a sustained release. So before I go into the effect, how does the effect looks, what I want to say is what is the efficiency of drug absorption, because you want to see what you want to reduce the application of drug. You want less amount of drugs and better effect. So you want a very good amount of drug to be absorbed on the surface so that you don't have to feed a person a kilogram of bacteria or bacteria board, something, right? So what I did, we did some testing analysis and try to analyze the efficiency of drug release. We did it in both ways. We did it in using spectroscopic, absorption spectroscopic techniques as well as HPLC, and we found the drug absorption efficiency when you applied around like 15 mg of drug per 100 mg of bacterial board in dry wet, actually it's around 67 to 70%, and drug release efficiency is around total drug level was 15 mg and we applied 20 mg. So yes, and the drug release amount is around 81% over a period of 15 hours. Okay, now how does it actually change the scenario of cancer treatment? If you see these animals, what do you see? This is a Swiss albino mice which is bearing a sarcoma 180 tumor. Now this is a control mice, and you see a huge lump of tumor over here. And then if you treat those mice with empty bacterial board, or even I didn't put another panel, or even lactobacillus, only lactobacillus, you don't see much change. Now the recommended dose of 5-fluorouracil, or rather capecitabine, I would say, is 50 mg per kg per day for 3 days for 3 doses. Okay, for over a period of 10 days. Now, so we used much lesser concentration of capecitabine, which is 25 mg, half of the dose, and what we saw that at a half of the dose you see a little more reduction than what we actually get without empty bacterial board. So that this terms means 5-fluorouracil 25 mg per kg dose. This term means a bacterial board loaded with 5-fluorouracil 25 mg per kg dose. So when you did that, we saw a little reduction of tumor. But when we use the recommended dose of 5-fluorouracil or capecitabine for oral delivery, we saw a fabulous result. We saw almost like 95% reduction of the tumor volume using empty bacterial board. So, and this shows day-wise change of tumor volume. Okay. Now we try to analyze whether this is actually just decreasing the volume or killing the tumor. Again, my again my slides are not moving. Can you please help?
Devjit, can you help? Devjit.
Yes, sir. Yes, sir. Yes, sir. I am working on it, sir.
Yeah. So this is just an H&E analysis. I'm not showing all the IHC results over here, but this H&E analysis shows that in controlled tumor or with antibacterial board, there is no necrotic region in the tumor. That means tumor is growing, they are fine, there are no dose. But when you use the drug which is half of the recommended dose, you see a quite a small bit of necrotic region in the tumor. And but when you are not using with antibacterial boat, but if you use it with antibacterial boat, you see a lot bigger region of necrotic tumor. And when you use a recommended dose, your volume of that necrotic region has increased. Now, what does that mean? That means only that tumor is shrinking, your tumor is not growing anymore. So we also try to see the effect of that bacterial boat on the life expectancy of the mice. So this is with ESE tumor. ESE is a liquid tumor which is you can put in the peritoneal cavity of the mice. It's a liquid tumor, so its effect is very fast. So mice dies or animal dies with this tumor at very fast. It can metabolize at a very fast rate. So if you see the control mice, this is our initial experimental data. So we did it to this try. We did it with 2 mice. So with control mice, the average lifespan after induction of the tumor is say around 15 days. With empty bacterial boats, it's same. With LR means Lactobacillus reuteri, it's almost the same. All has an average lifespan of around say between 10 to 15 days. But when you use recommended dose of lactobacillus 5-fluorouracil or capecitabine, the lifespan has increased. Almost like lifespan has become twice. The average lifespan has become around 25 days. But when you apply the same dose with antibacterial boat, the lifespan has become 35 days after induction of the tumor, which is fabulously well. It's have almost increased around 200%. So this system is actually very, very effective. So what we can conclude is when we from this data, when we put those particles on the bacterial board, on the bacterial surface and in living condition and allow the bacteria to get and go and bind over there in your intestine or GI tract, it can release the drugs from there very efficiently. I'm not showing that release kinetics data or pharmacokinetics data right now because we are some of the data are still pending. We have some data and some of this data we're still waiting for some of the data. But whatever data we got, we found an wonderful expression of wonderful release of drug over a period of for 5 capecitabine. We found over a period of we could detect drug over a period of around 6 hours for inside from the blood, whereas if you don't apply it through empty bacterial boat, you can detect by the drug for 10 minutes maximum from the blood. After that, it's all gone. So that shows we have increased the absorption or the bioavailability of the drug in the blood for over like 60 times, which is fabulous, because when you can get that much increase of the bioavailability of the drug inside the blood, you reduce the dose by many fold, which will definitely decrease the side effects. And especially when you're talking about anti-cancer drug or something, the reduction of side effects is very important. Most of the drugs are applied through intravenous region, intravenous route. Very few drugs actually get applied through your oral route because they have lower absorption rate. But with this formulation, probably we can change the scenario. We have already filed patent for this work, an Indian patent at this point. We're also working on filing an international patent, US patent actually. And so here I would like to show, this is my lab, and this is the girl who did all the work. She's Porman, and these are my other PhD scholars and few MSc students are there. I would also like to thank Dr. Biswarup Basu and his PhD scholar Shobhik Das from Chittaranjan Cancer Institute. Most of the cancer testing, like the animal data you saw, is done by Dr. Biswarup Basu. I would like to acknowledge Dr. Neha Garg and his heart scholar, Mr. Kundalik. They have done that fabulous work on the same. We don't have FECM facility over here. We're actually about to purchase it. Which it should be here if the COVID by now, if the COVID situation was not around. But anyway, so Dr. Neha Garg and Kundalik helped us with getting those same data done. Dr. Abhrojyoti Ghosh from Bosch Institute helped us for standardizing our biofilm assays. And last but here, Dr. Virender Garg is there. Dr. Virender Garg, with him, whom we are actually trying to go for the phase 1 trial and phase 2 trial. That's what we're working right now. Dr. Virender Garg is in PGI, Chandigarh, so we'll be applying this system very soon to actual human patients. And I would also like to thank DST for funding this project, as well as our department and the university for providing us the facility. I would like to go to the last slide, but the last slide is basically thank you for your patience. You have been a wonderful audience. I would be very happy to take any questions if you have.
So there is some question in our chat box.
Yes.
So,
Okay. Yeah, I got that. Okay.
so first question is from the Gurjan Singh, and his question
Okay.
is, what is the release kinetics of bacteria boat?
Okay. The release kinetics, we have studies using spectroscopic technique and HPLC data. I have already shown the data. Using fluorouracil, what we found is almost 80% of the drugs. So you see a fast blast. So within one hour, you see release of almost 40 to 50% of the drugs. But after that, you see a sustained release till 6-7 hours, and then sharp decrease, because by 6-7 hours, almost 70-75% drug get released with this current formulation. But you can always tweak the formulation a little bit and increase that. And we could, using our spectroscopic technique, we could actually detect the drug till 15 hours, which is almost like 85-86% of the drug's concentration together. If you take the whole drug concentration what we loaded earlier, that's like around 85-86% drugs which got released from there. Now, if you talk about pharmacokinetics, we could detect the drug inside the blood for 6 hours. The results is quite clear, because what we found in our in vitro data, we found the around 6 hours where you see like almost 70% of the drug get released. After that, you see a sharp decrease of drug concentration. So probably that is why we could detect the drug till 6 hours from the blood and not after that.
Okay, sir. The next question is from Karthik Digar, and his question is,
Okay.
how much long time the bacteria can live after encapsulation of the drug particle into the bacteria, and have there any duration that bacteria can live that much time in the stomach after encapsulation?
Well, that's a good question. So inside the stomach, we didn't check after. So till 12 hours we checked, because it's very difficult. So with the way, I'm wrong. Inside the stomach, we didn't check how long it can stick. The reason is it needs a lot of sacrifice of the animals. The way we did our earlier experiment is basically when a goat got sacrificed, we took the stomach from the butcher house and we cleaned it. We made it iodine sterilized and all, and then we incubated the bacteria boat and bacteria for 20 minutes with or without drugs, and washed it thoroughly using intestinal fluid, and for I guess twice, and then we did microscopic study to see whether those are attached or not, and we found a fabulous attachment. But when we did in vitro experiment, we found the bacteria are quite stable. They can grow. We did experiment for 7 days. So they can grow for even on 7 days. So they are alive, they are not dead. Obviously, after 48 hours, they start growing at a much faster rate. So that time, probably the particles become little unstable on the surface. They start falling off. So I have some results which shows that if you keep it more than 48 hours or something, the particle starts falling off. And after 72 hours, the rate of falling off of the particles are very high. With increase of time, the biofilm production also increases. So that's good enough to show that the bacteria is gonna stick to the intestine because they are live, they are metabolically active, and they are producing biofilm. So they should be able to stick to the intestine for quite long time. And we need the window, and these are the bacteria I'm talking about, which we already take, like it's medically prescribed bacteria. You take it when doctors gives you Bacillus subtilis or lactobacillus doses when somebody gets stomach upset or something. So these are good bacteria, very known to stick to the intestine. And according to our assumption, I didn't do it in vivo for time-dependent experiment yet, but that's a very good suggestion. We may plan. I would love to actually, if we get enough animal, because sacrifice of animal is again an issue. We have permission for limited number of animal to be sacrificed. But if we can get it, we'll definitely get it done, and we'll see how long in vivo inside the animal it can stay. I hope that solved your query.
Okay, thank you, sir. Now I like to request our honorable director, sir, for virtual handover of e-memento to Dr. Deeptiman Chaudhary, and as well as for the vote of thanks.
Once again, good evening, Dr. Chaudhary. Actually, after this Amphan in West Bengal, there is a lot of problem with internet. Even my internet is going and coming. There are some issues. You may not be able to hear me. Am I audible now?
Yes, you are. You are.
Somehow, I don't know, because these internet problems are there. But very quickly, I think Devjit, one more question was there. What are the potential toxic effects? Did you ask this question?
Yes, sir. There is some question. What are the potential toxic effect? Again, from the Gujran Singh.
Okay, that's a good question. So, 5-fluorouracil, unlike most of the other cancer, toxicity is less. Okay, when you apply it through oral route through capecitabine, because most likely its half-life is less. But it's not clear how toxic is that. But our system, so far, I would definitely say we tried to see the toxicity. We did a liver toxicity analysis, liver function test, kidney function test, liver function, kidney function, and we also checked different blood parameters, and we didn't see any change in toxicity. I mean, if you just apply your normal 5-fluorouracil versus 5-fluorouracil through BacteriaBoat. For the shortage of time, I didn't show that, but yeah, it didn't increase the toxicity in the blood for sure. And again, it's release kind. If you talk about the release kinetics, what is actually happening? You are releasing the drug at a much lesser rate in compare with just capecitabine. If you just apply capecitabine will show a very fast absorption. within you will see a increase of high peak of the drug within 30 minutes, and that means within 30 minutes you see almost 60 to 70% of total absorption, and within an hour you will not be able to detect anything. But with our system, because it's a sustained release formulation from the stomach, the release is actually less. And I'm talking about the same drug. I'm not talking about anything else. We didn't do any modification. That's why this system is beautiful, because it can be applied with any drug which can get absorbed without any modification, get absorbed by the bacteria. And it will release at a much slower rate. So toxicity, whatever it will have, will actually get reduced rather than it will get increased. So yes, we didn't see much changes. I will not say reduce, because pyrchlorouracil itself is not very toxic. But yeah, we didn't see much difference.
So other question from the Balakrishnan, that how the
Yes.
bacteria would eliminate after drug release.
So the bacteria both eliminates from the drug release. So again, it's very difficult to say that. It's just a normal bacteria. If bacteria goes colonize in the intestine, and then they stays there. If they dies off, or when the colony become bigger with friction of food and other things, they just fall off from the surface. So till it's not dividing for say around 48 hours, or at least for 24 hours, it would be very stable, because it is producing a lot of biofilm. Once it will start dividing, then it will become a little unstable, and maybe after 72 hours or so, it's just fall off, just like any other bacteria. But again, we don't know, because it's very difficult to detect bacteria from feces. Like the difference between a lactobacillus and a lactobacillus bacteria both, it's very, very difficult. We tried, but we couldn't differentiate that whether we have. I mean, we got bacteria definitely. Maybe we can. We haven't checked for sequencing, but it's, when you're talking about live animal, you already have some kind of lactobacillus and other bacteria inside it. So with MRS media, it always, when you take it and put it in the media, try to do OD checking and all, you still get some kind of bacteria over there, because it comes with their food and all. So it was difficult. But I guess that's a good question. We'll try to do it again. We'll see how can be managed. But at this point, I'm not been able to say how it will follow.
Okay, thank you, sir. Back to Director sir again for the felicitation and the vote of thanks.
It is really very unfortunate that today somehow I am unable to connect properly whenever it is required. But I am trying my level best. I think I have kept my video off so that I can be audible a bit. So thank you very much for your very elaborate discussion on this. Whatever amount I have heard that you have told in a very lucid way the research you are doing, particularly the way you described this 5-fluorouracil and others. There are some more questions. It seems one Mr. Gurjant Singh is quite interested in doing some research. I don't know what is his background, but I will request Gurjant Singh to mail the organizer whatever other questions you have.
Yeah, you can easily share my. Sorry, sir. You may share my email ID to Mr. Gurjant Singh.
That is whatever mails we'll get, we can share it with you. directly, and so that you can answer them back to the person who is interested to learn or to know. So once again, on behalf of Guru Nanak Institute of Pharmaceutical Science and Technology, I convey my heartfelt thanks to you, and I would definitely want and definitely like to have in future some more interactions with you, some more seminars with you, because in fact I was not very satisfied because I could not hear the whole seminar, and still I think I am audible, but I'm unable to see anyone, or rather I think no one is able to see me. So whatever the matter, I will definitely request you that in future we should have a chance to interact with you in furthermore seminars. We are also planning some online conferences in future within a month or two, some international conferences, and I would also like to have you as a speaker for those kind of conferences which we will be doing.
Sure, that will be. I'll be honored to do that.
So that's a very good honor for us also that you be with us. Thank you very much once again, and if
Thank you for hosting me here. It was wonderful audience.
I suppose you are basically from where? Native to Calcutta?
I'm basically from Kolkata. Yes, I'm. My house is at Thakurpukur. So I'm actually we did the PhD from the same lab as Dr. Ghosh Dostidar, Devabrata Ghosh Dostidar. He is my lab junior.
That's very nice. That's very nice. So whenever you are coming to Calcutta, I would also be glad and honored if you sometimes do visit our institution at Shodhpur.
Sure, sure. I'd love to do that.
Thank you very much, sir. Thank you once again.
Thank you.
And
Thank you for it.
Memento, which that is only a e-memento, it will be mailed to you. But
That's
anyway,
absolutely
I can't see what Devabrata has made, but definitely it will be emailed to you. Thank you, sir, and namaskar.
Thank you. Thank you.
Thank you,
Okay,
everyone.
guys.
Nijit, if nothing else is there, we can end the.
Okay, thank you, everyone, and we appreciate you are being here. Thank you again for joining us. And this is to inform all the participants that they will receive their e-certificate within 7 days in their registered email ID. Once again, thank you all.
Thank you, guys. Bye.
Thank you.